Welcome to Lecture 11 on the topic of plant growth and development. This lecture has been delivered in two parts, and this is the second part. Please ensure that you watch part 1 before continuing with part 2. This lecture is part of the topic Plant Physiology, which is offered in the Bachelor of Agriculture and Technology at North Melbourne Institute of TAFE. For more information about this subject and the other courses that we offer, please visit our website at www.nmit.edu.au. My name is Dr. Nikki Cooley. In the first stage of this subject, we looked at individual components of a plant and took a virtual tour throughout the plant. As we were taking this tour, we learnt about the very important physiological components. Many of them aid in plant growth and development. In this lecture, the emphasis will be on the whole plant responses to plant growth and development. In this lecture, we will look at the specific components of root and stem growth. The root tip has 4 developmental zones. In Zone 1, the root cap. This contains a unique set of initials that covers and protects the apical meristem. It also functions in the perception of gravity, also called gravitropism, and the secretion of compounds. In the developmental zone 2, the meristematic zone lies just under the root cap. This contains the initials that divide and differentiate into various tissues of the root structure. In the third zone lies the elongation zone. This is the site of rapid and extensive cell elongation. And finally, the maturation zone. This is the region where cells become differentiated after division and elongation has ceased. Organs such as the lateral roots and root hairs may form at this stage. The figure on the slide shows a visual representation of the root tip structure. You will note the lateral root prydomium, the mature zone, the elongation zone, the meristematic zone and the root cap. All of the important components of each of these zones are illustrated. The origin of the root tissues can be traced back to specific initial cells in the root apical meristem. At the centre of this region is the quiescent centre, or QC, named for its low rate of cell division. 4 distinct sets of initials can be defined in terms of their position and the tissues they produce. The Columbia initials, the epidermal lateral root cap initials, the cortical endoderm initials, and the stele initials. The Columbia initials divide to form the central portion of the root cap cells. Epidermal lateral root initials divide to form lateral root cap cells and epidermis. The cortical endoderm initials form the cortex cells and the endodermal layers, while the stele initials form the vascular system and the pericycle cells. The figure on your screen shows the root initials. On the left-hand side, you have an image taken from a root tip. On the right-hand side, you have a schematic drawing of where all the initial components are located. Let us talk about auxin and the role it plays in the root apical meristem. It is believed to play an important role in positioning of the root apical meristem and guiding its behaviour. The position of the QC coincides with a maximum concentration of auxin. Models show that auxin moves down from the shoot through central regions of the seedling and concentrates in the QC. Auxin is then transported outward, then inward towards the shoot. The figure on the slide summarises some of the experimental evidence demonstrating auxin movement. This has been done by combinations of method, looking at pin protein distribution, modelling of auxin flow, and DR5 to GFP expression. Studies suggest that cytokinin acts in opposition to auxin. Auxin is synthesized in the shoot and transported to the root, while cytokinin is synthesized in the root and transported to the shoot. Auxin promotes root development, while cytokinin suppresses roots while promoting shoot development. Cytokinin begins early in the hypothallus of the glandular embryo. On division of the hypothallus, cytokine expression is lost in the basal cell but retained in the apical cell, which divides further to form the QC. The following image is an SEM of a shoot apical meristem. This shows the very early stages of initiation. You can see regions P1 through to P4. Now I'm going to talk a little bit about zones and layers of the shoot apical meristem. The central zone, or CZ for short, contains a cluster of infrequently dividing cells, similar to the cells of the quiescent center. The peripheral zone, or PZ, divide more frequently to produce cells of lateral organs such as leaves, while the rib zone, or RZ for short, contains cells that give rise to the internal tissues of the stem. The image on the slide shows a high-power microscopy of the zones and the layers of the SAM, the shoot apical meristem. As we described in the previous slide, you have the central zone, where the cluster of infrequently dividing cells occurs. The peripheral zones, either side of the cluster zone, and the rib zone. These cells contain and give rise to internal tissues of the stem. So the shoot apical meristem has cell layers, and these cell layers contribute to different or specific tissues of the shoot. The L1 is the outermost layer, and this produces the epidermal layer. Cell divisions here are anticlinical. The L2 layer has the same type of cell divisions, that is, they are anticlinical. And this layer produces the subepidermal layer of tissues, while the L3 layer is the most inner layer, and this generates internal tissues. But you will note that this L3 layer cell division is quite different. Rather than anticlinical, it has random orientation of cell division. So you will note L1 and L2 layers have very structured division, while L3 has a very random orientated division, and this helps shape the form of this tissue. The shoot apical meristem has cell layers that contribute to specific tissues of the shoot, as was discussed in the previous slide. In the image on this slide, you can see the location of the L1, L2 and L3 in the shoot SAM, the shoot apical meristem. In the figure on the left-hand side, you will see the different types of cell division that are indicated. These overlay where the L1, L2 and L3 layers are occurring. The architecture of the shoot mainly depends on the production of lateral organs, leaves and branches. Shoot systems also increase in complexity by the outgrowth of lateral roots. During leaf initiation, the characteristic arrangement of leaves on the shoot is termed as phyllotaxy. There are 3 basic phyllotaxy patterns, and these are represented on the slide. These patterns are formed after the initiation of leaf primordia on the shoot and the apical meristem. It will be no surprise for you to hear that leaf primordia initiate where auxin concentrations accumulate. The image on the slide shows a visualization of auxin accumulation, the site of primordia cell division, and how the order of primordia division is correlated to the auxin concentration. As auxin concentrations increase, primordia elongates. Leaf geometry can be described by a proxial distal axis, base to tip, a lateral axis, that is from one edge of the leaf to the other, and an abaxial to adaxial axes, which is the upper to the lower surface. Mechanisms by which leaf patterning occurs remain unclear, although several important genes have been identified. The phantastic mutant fails to establish leaf patterning, and the leaf forms a cylindrical shape. Here we show a visual representation of this mutant. We start with the wild type that has both adaxial and abaxial. This leads to inductive interactions between the distinct layers. Lateral growth follows, and you can see abaxial and adaxial growth. While in the mutant you only get abaxial growth, and this results in a filamentous shape as opposed to a laminar shape. Now we will examine lateral root growth in more detail. The first signs of lateral root initiation are periclinal divisions of a small number of cells in the pericycle. The new root tip eventually ruptures, and the endoderm and cortical layers of the parent root. A new root apical meristem is established at the tip of the lateral root. Lateral root patterning is influenced by auxin concentrations as well as environmental factors such as nutrient availability and soil moisture. On the slide you will see a visual representation of root development in the number of processes that we have described. These can be separated into 5 distinct stages. In the next series of lectures, we will look at plant hormones and their interactions. I am going to just touch very briefly on growth and development and hormones in this slide. This image is from a paper, Phytochromes Regulate All Aspects of Plant Growth and Development. It is from a paper published in Nature by the authors Santer, Iran, Cardon Vascular and Estelle. It shows you where the plant's hormones are regulated with respect to the plant. The plant that they're showing here is Arabidopsis thaliana. As you can see, auxins, gibberellins, ethylene, jasmic acid, brassindone, salicylic acid, stringophone and abscisic acid and cytokinins are all involved in growth and development. The table on the slide is a comparative account of plant growth regulators on plant development. On the left-hand column, you will see listed the stages of growth and development, from root development through to dormancy. The following columns after this represent whether the hormone is present with a cross or absent with a slash. The hormones that are examined in this comparison are auxin, gibberellin, cytokines, abscisic acid and ethylene. Please understand that a couple are missing from this diagram, but it does allow a very simple and quick summary of which hormones are involved at which stages of plant development. Could you please read the recommended textbook Plant Physiology by Tays and Zeiger, 5th edition, Chapter 15, section on the structure and synthesis of plant cell walls, page 314, the section on the pattern of cell division, Chapter 15, page 328 to 335, and finally the whole of Chapter 16 on growth and development, pages 339 to 374. Please make relevant notes and insert your notes into the lecture at this point. And that brings us to the end of the topic of plant growth and development. In part 2, you have understood now about meristemic tissues, root apical and shoot apical meristems. You've understood about how leaf shapes come about and the patterns of leaves. You have an idea about lateral root growth. And, importantly, you understand about the role of hormones in plant growth and development, the two important being auxin and cytokinin. This is the end of part 2.